Hello and welcome to Biochemistry lesson, high level edition. So today we are going to be talking about cell respiration again, but we are going to go further from this previous class where I showed you glycolysis. So in this one here, we saw that glucose, a 6-carbon molecule over here, is broken down into 2 3-carbon molecules over here, pyruvates. In this process, we produce ATPs. So we have here 4 ATPs produced, but 2 have been spent. So we produce 2 ATPs overall. And we produce also some NADHs over here, NADHs over there. So we got some electrons from that reaction. So this here is the glycolysis, what happens in preparation for what you're going to be having today. Glycolysis happens in the cytoplasm. So 2 pyruvates in this chemical reaction have been generated inside the cytoplasm, produce 2 ATPs, produce 2 NADHs, and now we are going to carry on. So here we have the pyruvate. So pyruvate is a molecule that's half a glucose roughly, but is still pretty energetic. We just broke it in half and separate the energy that was there, and now we are going to be able to put that in a link reaction, also known as acetyl coenzyme A reaction, and the Krebs cycle or citric acid cycle to squeeze some more ATPs and other stuff out of it. And all this is going to be happening now inside the mitochondria. So now pyruvates enter the mitochondria to be able to carry on the chemical reaction. So let's take a look in the beginning. So when pyruvate enters the mitochondria, in the matrix of the mitochondria, in the middle there of the mitochondria, it's going to be there 3-carbon and it's going to be decarboxylated. So the first thing that is going to be happening is that those 3 carbons are going to lose 1 carbon dioxide, so it's going to be losing 1 carbon, and it's going to give electrons in that process. So now electrons are stolen from this chemical reaction and they are going to be added into a NAD to make a NADH plus H plus. So basically we have 1 more electron here per pyruvate. In this reaction is going to be 2 NADHs per glucose, because this here happens twice for every glucose. And in this process that we stripped 1 carbon dioxide out of the pyruvate, we will end up adding a coenzyme A, which is a transporter, to be able to carry those 2 carbons over there, which is called an acetyl. That acetyl is going to be with the coenzyme A and is going to be ready to go into the next step. So this process here made no ATP, but made some NADHs and got the acetyl ready, highly energetic, to go to the next step. So we produce 1 NADH per pyruvate, 2 NADHs in total, 1 carbon dioxide pyruvate, 2 carbon dioxides in total per glucose. So 1/3 of the carbons in a glucose so far is over. They have been produced. Okay. After the link reaction or acetyl coenzyme reaction, now we are going to get, so this is for the ones that like to see pretty images. So you take a look here on the formula, the pyruvate, the acetyl here, very good, have fun. So after the link reaction, we go to the citric acid cycle, also known as the Krebs cycle, which is the nightmare of anyone that have ever studied biochemistry in college. So if you know anyone that studied biochemistry in college, you ask, have you studied Krebs? And they're going to go, whoa, because no, it's the nightmare. However, it doesn't need to be the nightmare. No, Krebs cycle, okay, it's central, very, very important. Everybody that studied biochemistry has to study that. But we don't need to memorize everything and know exactly every single molecule that goes in there. It's very important to understand the concept, what's going on over there with this Krebs cycle. In the same way that in the link reaction, what went on was that we broke down the pyruvates and took away 1 carbon dioxide, and we made a little bit of electrons in that end that went to the electron carrier, the NADH. So what's going to be happening now is that we carry on. That same acetyl coenzyme A, 2 carbons, are going to be carried through the citric acid cycle, and it's going to be entering in that place over there. We have in the cycle already a 4 carbons molecule. We put 2 carbons coming from the acetyl coenzyme A, we make a 6-carbon molecule, and the coenzyme A then goes away to bring another acetyl to this reaction. In this process, we made 4 plus 2, we made a 6-carbon molecule. So if you have 6 carbons molecule, we are going to be able to strip a carbon dioxide out of it. And when we do that, electrons are released and we can make one more NADH per pyruvate, per acetyl in this case, making a 5-carbon molecule. But if you have a 5-carbon molecule, I can also strip one more carbon dioxide, which will give away electrons and protons, which I can use to make one more NADH per pyruvate, per acetyl that's in there. And then I make a 4-carbon molecule. However, this 4-carbon molecule is very different from that carbon molecule over there, that 4 carbon molecules. It can still be transformed, it still can be stripped a little bit of energy. So that's what's going to be happening now. There is an extra amount of energy over there that will be used to produce one ATP. So bonding an ADP plus P into ATP. Yay, we made one ATP per pyruvate, 2 per glucose. And we are also going to have some electrons can be taken, put into a FAD to make a FAD-A2. A FAD is a fancy NAD. So for our purpose here, think like that being kind of a NAD. And we also still have electrons in that 4-carbon molecule that's still not the one that we started with over there, that are going to be stripping some more electrons. Then we're going to have now making one more NAD. So in this entire process here, we started with a 4-carbon molecule, put into making 6, 5, 4, 4, 4, and we are here in the beginning again. This is the Krebs cycle, because it starts in a place, go back to the same place. I add 2 carbons here, but I get out 2 carbons over there. So this is the process. All this here goes 1 round for every pyruvate. So for every glucose, it's going to be 2 rounds. So in here, you saw that we made 3 NADs, 1 FAD, 1 ATP, and 2 carbon dioxides, all this times 2. So in the end, just on here, we have made 6 NADs, 2 FADs, 2 ATPs, and 4 carbon dioxide. And at this moment, because I made now 4 carbon dioxides over here, I had stripped 2 out of the glucose of the pyruvates in the link reaction. Now I stripped the remaining 4, my glucose is over. The glucose totally over, has been consumed, I exhale it out, and now I have made 2 ATPs in glycolysis, 2 ATPs in the citric acid cycle, but I still have stuff that are highly energetic, which is the NADHs and the FAD-A2. So with the NADH and FAD-A2, I will be able to extract even more energy to make ATPs in a future step. Okay? So if you got the idea of the citric acid cycle, very good, that's it. Otherwise, let's discuss it in class to make sure that you get the idea. This is for the chemistry freaks, people that like chemistry and formula and all that stuff. We have here all that we saw before. So we have the acetyl coenzyme A is entering here. So we have a 4-carbon molecule there, the oxaloacetate, becoming a 6-carbon over here, like citrate. You don't need to know the name of this, you don't need to know the formula. This is there just for the ones that say, oh, I want to see the formula, I want you to see what are they made of. So we have that over there for them. Okay? As we are talking about stuff that happens inside the mitochondria, it's important that we have an idea of the mitochondria structure. So this here is the mitochondria, a diagrammatic view of it, where you have an outer membrane, an inner membrane. So let's see everything that you have here. Outer membrane is an outside layer, and we have an inner membrane. By the way, these 2 membranes here support the idea that mitochondria are actually bacteria that through the endosymbiosis entered another cell and became one of our organelles. Very, very important. Changed the history of life. Okay, we have outer membrane, we have inner membrane, so therefore we have an intermembrane space. The intermembrane space is a very narrow space, and because it's narrow, it's going to be very important in the next step. Okay. Additionally, the folding of the intermembrane space, we call it a crista or cristae, which is all folded, increasing the surface area of the inner membrane. And the inner membrane have many proteins over there to perform the function of the mitochondria, so a bigger surface area make it work much better. Okay, although mitochondria has 2 membranes, an outer and an inner one, it has a much bigger surface area for the inner membrane than it has for the outer membrane. Additionally, we have here what would be the cytoplasm of a cell, In the mitochondria, we call it the matrix. Okay, the matrix is the cytoplasm of the mitochondria, but we don't call it cytoplasm because it's not technically a cell, it's an organelle, so we call it the matrix. Okay, you also have kind of 70S ribosome. You have prokaryotic ribosome, which is another evidence for the endosymbiotic theory, and the mitochondria, therefore, is capable of producing its own proteins. Okay, it also has its own circular naked DNA, genetic information, which also supports the endosymbiotic theory. This is actually a bacteria living inside of us with its own DNA, ribosome, doing all its stuff, and two membranes to support all that idea. Okay. Later on, when we finish seeing all the cell respiration, it's going to be very interesting if you are capable of associating the structure of the mitochondria with how it performs its function. For example, how it does the link reaction and the Krebs cycle. It has proteins in here in the matrix that are produced by the 70S ribosome with information from the endoplasma, from the circular naked DNA, and therefore it's capable of producing all those chemical reactions, all those proteins, enzymes and stuff. So the fact that it contains its own 70S ribosome and circular naked DNA explains the idea that we have many chemical reactions happening in there. Okay, so another image here. This is a little bit more realistic, showing that the intermembrane space is very narrow because this crystal actually goes very, very packed together. Okay, a real image on the right showing the crystals and the intermembrane over there. With a diagram on the left. So this is a mitochondria. Okay, the powerhouse of the cell, the place where after glycolysis we are going to have aerobic cell respiration. Okay, I hope you have enjoyed this class, and take a look on the quiz now so you can exercise what you've learned so far, and I'll see you in class for us to have a discussion. Cheers, bye bye.